Good morning, everyone. Let's come to the important topic of molecular biology, which is proto-oncogenes and tumor suppressor genes in cancer cells. So, what are proto-oncogenes? Are those genes whose products promotes cell growth and division. They are not generally, oncogenic, but once it has been mutated, okay, that becomes a problematic or oncogene. Okay, this gene encode a transcription factor that stimulate the expression of other genes, a signal transduction of molecules that stimulates cell division, and single cell cycle regulators that move through the cell cycle. So what's an oncogene? Is a proto-oncogene that is mutated, right? So the proto-oncogene, when it has been mutated, it becomes an oncogene, okay, and abruptly express and contribute to the development of the cancer. So you would find here there are different types of proto-oncogenes. What you are finding, for example, c-Myc, okay, which is an important in transcription factor and regulates cell cycle differentiation and apoptosis. So how it is altered in the cancer? You see the translocation and amplification or point mutation, what you find, and which cancers is associated as lymphomas, okay, and leukemias or lung cancers or many types. Again, you have different types of proto-oncogenes, for example, cyclins, so which binds to the cyclin dependent kinases for CDKs and regulates cell cycle. It's important in gene amplification and overexpression. And what cancers it is associated with is, for example, lung cancers, esophagus cancers, and many other types of cancers. Okay, so what are different types of genes? Another type of gene, what's called tumor suppressor genes. So you will find the tumor suppressor gene normally regulates cell cycle checkpoints and initiate a process of apoptosis. So it is like guardian. You have to guard the genome in such a way so that it's not undergoing sort of oncogenesis. Okay, so when tumor suppressor genes are mutated or inactivated, the cells are unable to respond normally. Okay, and there hence is there it needs to have a such kind of checkpoint to undergo apoptosis, so as DNA damage doesn't occur. So there are certain kind of genes, for example, p53 genes, for example, or RAS genes, which includes signal transduction molecule that are associated with the cell membrane. So here you see there is a growth factor, okay, which is binding to the receptor. Now inactive RAS changes to the active RAS, okay, and this RAS is it activates the cascade of protein, which activates the MAP kinase, which activates the transcription factor in such a way to express certain kind of gene. So there are different types of RAS, for example, lysine RAS, what's called KRAS, right? So mutation that convert RAS proto-oncogene to the oncogene freezes the RAS protein to its inactive conformation and constantly stimulating the cell to divide. So RAS proteins is actually a proto-oncogene, and when you have a RAS which has been mutated, okay, then that can be a particularly dangerous in the cells where the signaling is involved. Then you have one more important factor, which is almost a celebrated gene. What you will find is a p53 gene or tumor suppressor gene, which is a p53 gene which encodes for a nuclear protein, and that acts as a transcriptional factor, right? It's a p53 is a transcriptional factor and represses or stimulates a transcription of more than 50 genes, actually more than 50 genes, right? So p53 is involved in continuously synthesized and rapidly degraded, thus present at the lower level. So increased levels results from the increase in the protein phosphorylation, acetylation, and post-translational modification. So what you see in p53 is in the stressed cells, unstressed cells, then the MDM2, what's called murine dependent double minute 2, which is binding to the TAT domain, and there's a ubiquitin, which then there is a p53 degradation. But when a cell is sensing certain kind of any environmental insults, including nutritional deficiency or any kind of xenobiotic substances, what happens is, so the MDM2 is released and there's a phosphorylation which is happening. Now this phosphorylation, okay, signals up p53 to be active. So once it has been active, then it activates p21 to stop the cell cycle and to undergo apoptosis, what called J1 SGS checkpoint. Then other proteins called BAG gene, which is apoptosis, and DNA repair, so GADD 45, which is activated, and p53 control, okay, to activate p53, right? So p53 can arrest the cycle at several phases, okay, and the cell cycle p53 are unable to arrest the cycle checkpoint and enter apoptosis or response to DNA damage, so they cannot do that, and such kind of problem you will find in the cancer. For example, a loss of mutation of both alleles of RB1 tumor, okay, tumor suppressor gene, it contributes to many cancers due to unregulated progression through the cell cycle. Again, what's the another classical example what you'll find in the cancer, like familial retinoblastoma, or where you are finding RB1 is generally is mutated from parents to the offspring, or sporadic retinoblastoma, okay, where mutational events occur in both the cells. So all these are the cases where you find that the p53 has been mutated. Again, you will find another protein, or as I was saying, retinoblastoma, RB1 protein. Again, it's a tumor suppressor, as what you find in the p53. Again, it's control the G1S cell cycle checkpoint and preventing passage into the S phase. So in many cancer cells, including retinoblastoma cells, are copies of RNA RB1 gene are defective, inactive, or absent, which is making it progress to the cell cycle, and it's not regulated. Okay. So the problem with all these mutations, the p53 genes or retinoblastoma gene have a different kind of effect, which is you find. For example, you see most of the cancers result from somatic mutation, but 50 forms are hereditary cancer, 1 to 2% are known. For example, if you are finding BRCA1 which has been mutated, right, so you'll find breast cancer, and APC, you'll find polyposis, or when you find a RET, RET, for example, you'll find endocrine neoplasma. So these genes have been marked for different types of diseases in such a way. If this mutation is happening, then it's causing certain kind of tumor. I hope you understood the basic concept of tumor suppressor genes. Thank you so much for your attention.